All right, good afternoon, everyone. Thanks for coming out for the second of our Ecology Center seminar, Ecology Center seminars for the month. It's my great pleasure to introduce our speaker for the month, Dr. Malin Pinsky. Comes to us from Rutgers University in New Jersey. Malin grew up in Maine, where he fell in love with ecology tide pooling and got his bachelor's in biology from Williams College before moving on to Stanford, where he got his PhD while working in the Hopkins Marine Lab. He was then a Smith Conservation Research Fellow at Princeton before starting his current position as he's currently an associate professor in the Department of Ecology, Evolution, and Natural Resources at Rutgers. He's also a member of the Institute of Earth, Ocean, and Atmospheric Sciences. And he's received many awards throughout his career, including but not limited to being an early career fellow through ESA, an Earth Leadership Program Fellow, and most recently being announced as a AAAS Fellow. And without further ado, I'll hand it off.
Thanks so much, Tim. It's been a wonderful couple of days here. I just want to thank everyone for many fascinating conversations over the last day and a half, and for everyone for being so welcoming. A special thanks to the students for being incredible hosts. It's been a wonderful visit so far. So many of you heard me say yesterday that I focus on global change and understanding how and why biodiversity is changing. Trained in quantitative ecology and population genomics. And some of you heard my talk yesterday about ecological change that's happening all around us. And it's quite a gift in some ways to be able to give a second talk about the other theme of research in my lab, which is trying to understand the evolutionary change, often silent and in many cases unnoticed, that's also likely happening all around us as well. So we know that environmental change is rapidly exceeding historical levels of variability, whether that's climate change or habitat loss or harvest or invasive species. And a big question is whether, where, and when the mechanisms that species use to adapt to these changes will continue to be effective in the face of this rapid change. And species have a range of mechanisms for adapting to this change, including moving to new locations across a range of spatial scales, or persisting in place through behavioral adaptations, physiological tolerance, acclimation, or evolutionary adaptation. And a lot of the research in the global change field is focused on physiological tolerance or large changes in movements across space, ecological adaptation. That's what my talk yesterday was about. There's been much less focus on evolutionary adaptation and the role that evolution may play in helping species cope with rapid environmental change. You know, one explanation for why evolution has not gotten as much attention is that the traditional view is that evolution happens relatively slowly, and therefore we wouldn't expect it to be important in the face of the rapid environmental change that's happening. Another potential explanation, though, is that we just haven't had the tools for detecting evolution that is happening all around us. And in effect, it's been invisible. So there are a few key evolutionary processes to think about. One is genetic drift. So when population sizes get smaller, genetic drift becomes stronger. Genetic diversity tends to be lost. That reduces adaptive capacity, and at the extreme can also reduce individual fitness through inbreeding. On the other hand, there's also selection that can enable adaptive evolution, such that phenotypes in a population or a species change rapidly and continue to be suitable and have higher fitness in the new environment. Gene flow is also a key process, moving genetic alleles between populations and across space. And that can be adaptive or maladaptive or neutral. And all of these processes obviously interact with each other as well. So one key idea is the concept of evolutionary rescue, laid out theoretically about a quarter century ago by Tomolkovitz and Holt. And here we're looking at a case where a population the environment has just changed for a population. Its fitness has declined, and now population growth rates are less than 1, and it's declining to extinction. On the other hand, the population may initially decline, but then evolve, have higher fitness, and increase again. And that would be a case of evolutionary rescue. So these evolutionary processes operate in all realms. But as you may have heard me talk about yesterday, I'm also interested in thinking about how these processes may play out similarly or differently across different realms. So I just want to point out some of the differences between realms that may be important as we think about the likelihood of evolutionary rescue and other evolutionary responses. One just has to do with the abundance of species and the abundance of individuals within species. So apparently chickens are the most abundant vertebrate on land, about 20 billion chickens around the world. At least a small fraction of them wear sweaters. It's a lot of chickens, but it's actually a very, it's relatively small abundance compared to some species of mesopelagic fishes with about 4 orders of magnitude higher abundance. With large abundance comes very weak genetic drift, stronger selection, higher standing genetic variation, probably greater capacity for evolutionary adaptation. Another difference is often dispersal and scales of dispersal. So land plants often are dispersing over scales of like 100 meters to maybe a kilometer. Marine algae, we're talking about maybe 1 to 10 kilometers. But for many marine animals, it's often scales of sort of 10 to 100 kilometers. So larger scales of dispersal. That's important for understanding the scale of local adaptation and the demographic coupling between populations as well. The final point I just want to mention is that the kinds of human impact also differ in many ways between realms, habitat loss and transformation now being a really important factor on land, but direct human harvest still being a dominant human impact in the ocean, especially through industrial fishing, though habitat transformation becoming more important, especially in localized parts of the ocean. So as we start to dig into these questions, the tools of what are called temporal genomics provide a very powerful window into the evolutionary processes that are happening. Temporal genomics is just the application of repeated DNA sequencing to a population through time. It might be using museum samples and contemporary samples or other natural history collections or intentionally designed scientific studies. There are also a number of studies out there that are called evolve and resequence, where they're experimentally evolving a population and resequencing it through time, often done in the lab. But I think one of the exciting opportunities now that especially the costs and methods of DNA sequencing have dropped so much is taking these methods out into the wild and using them to understand evolution in the complex natural environment. These methods also allow us to focus on the relatively recent past and rapid evolutionary changes over a couple generations to a few dozen generations. These are also the timescales where we have relatively larger sample sizes in many cases, and also where we have relatively good environmental records that in many cases can provide the basis for stronger inference. So evolution at its core is a change in allele frequencies, and the patterns of change through time tell us something about the processes that are driving that change. So here we've got allele frequency from 0 up to 1, 100% on the y-axis, across generations here on the x-axis. In gray, we have alleles that are just drifting neutrally, not important for the fitness of a population. In blue, we have an example allele that has appeared and is strongly beneficial, so it rapidly increases in frequency to fixation. And then in red, we have some examples of deleterious alleles that have appeared, but they're selected against and they rapidly decline to extinction. So temporal methods are especially useful because many of the traditional methods for detecting selection across the genome are not as effective for the kinds of evolution that we expect to be important in response to global change. So as one example, hard sweeps are a case where a new mutation arises and quickly increases in frequency, like that blue allele I showed you in the previous example. In these cases of a hard sweep, they leave a distinctive footprint in the genome. So if you look at distance in the genome away from a site that has experienced a hard sweep, we see that diversity increases the farther away you go, or conversely, close to a site that's experienced a hard sweep, we have a decrease in diversity. This is across a few different diversity metrics from a set of simulations. On the other hand, we can also have what's called a soft sweep. This is a case where the genetic variant is already present in the population, but then the environment changes and that allele is now suddenly beneficial. In that case, it can increase in frequency, but leaves very little signature in the genome because it's been unlinked from a lot of from the variation around it. There are some methods that are starting to tease out hard versus soft sweeps, but still much harder to detect. In this case, though, the temporal signatures of allele frequencies through time provide a stronger and more direct method for inferring these kinds of changes. Another distinction I want to make is between a complete sweep, where the allele sweeps all the way to fixation, or an incomplete sweep, which may be actually more common in many cases of global change where evolution is still ongoing. So where the sweep has not, the allele has not increased all the way to fixation. So with this background, I want to provide a few short introductions to some of the projects where we've been applying these methods and some of what we've been learning. The first is a case where bats have been infected with a novel pathogen. Second is Atlantic cod, so a very intensively fished species in the ocean. Has supported many economies, especially around the Atlantic and parts of the Pacific. And then I'll end by talking a project that's still in progress, but starting to try to generalize beyond individual case studies to understand what characteristics make some species more likely to evolve and others less likely. So on this first case, this first story, white nose syndrome, or pseudogymnoascens destructans, is a fungus introduced to North America about 15 years ago. Bats are infected often during hibernation, and bats that are infected tend to wake up during hibernation, possibly because they're getting dehydrated. But when they wake up, their metabolism goes up, their energy use goes up, and they tend to burn through their fat stores during a time of year where they can't get more food. So many of them either starve or get dehydrated and die. So white nose was first identified in a cave in New York in 2006. It was likely introduced from Europe, where the disease is endemic, and actually bats don't seem to die at particularly high rates in response to white nose syndrome. But it's now spread across much of the US. So we have early sightings in purple and blue, more recent detections in red. We see it's spread quite rapidly. It's been detected in 21 species of bats, and some populations have been entirely extirpated. Some species have declined in abundance by about 95%. So it's been incredibly destructive. Large conservation concerns for many species of bats as a result of this novel disease. On the other hand, there are some populations, intriguingly, that are actually increasing now. So this is little brown bats, Myotis lucificus. And these are population dynamics for two populations. In New York, we've shown in yellow, we see a rapid decline, but then actually a partial recovery in recent years after infection, where this vertical line is when white nose was detected. And then in New Jersey, we also have a very rapid decline, but then a stabilization, and actually population growth rates now are just above 1. That's based on marker capture research that a good colleague of mine, Brooke Maslow, has been doing. And one of the things that's intriguing is that these cases look a little bit like what you might expect from evolutionary rescue, with these declines and then at least partial recoveries. So to test that idea, I'll present some work that was done by Sarah Genu-Wolfson, so a postdoc in Brooks' lab and also with me, and who's now just started a faculty position at University of Massachusetts, Lowell. And we collected tissue samples from the New York population, actually just initially during that initial mortality event. So 22 of the non-survivors. And then about 2 bat generations later, around 2016, collected another 21 bats from this particular hibernacula. These were wing punches. And then similar sample sizes from a hibernacula in New Jersey. So we did whole genome sequencing on these samples, about 53x coverage across the genome at a population scale called probabilistic genotypes, and for about 31 million single nucleotide polymorphisms, or SNPs. A SNP is just a single base pair location within the genome. So the first question was whether the historical population just died out and got completely replaced by bats from somewhere else that might appear as the non-survivors grouping entirely separately on this principal components analysis from the more modern populations. But when we actually look at the genomic data, we see that both the survivors and the non-survivors are grouping together, suggesting that these are same populations through time. FST is a metric for measuring the genetic divergence between populations. We're looking at about 2% divergence through time between these populations. So relatively small amount of genetic change at a genome-wide level through time in both of these hibernacula. But that's on average across the genome. We can also look at something called Tajima's D statistic. It tells us something about the frequency of rare alleles in the population. And one of the things that we see in the New Jersey population, so the one that declined to lower abundances and has stayed low for longer, is this classic signature of a population bottleneck, where Tajima's D shifts towards more positive values. And that's a sign of a loss of the particularly rare alleles in a population. And we actually don't see that signature of a population bottleneck in the New York population, where abundance has stayed higher and there's been more recovery. That seems to match up with the population dynamics of these two populations. We are especially interested, though, in testing whether there may be parts of the genome that are experiencing selection from this novel disease or other environmental changes. So if we look at allele frequency changes, these are averaged across the New Jersey and New York populations, across the bat genome. Each of these gray bands is a contiguous section of the bat genome. We call those scaffolds. And what we see is up top here, a number of alleles have changed frequency quite a lot, actually more than 50%. So really dramatic changes just over a couple generations. I should say that each dot here is a single nucleotide polymorphism within the bat genome. We can then use Wright-Fisher forward simulations to test whether the allele frequency changes that we're observing are consistent with what we expect just from neutral genetic drift and sampling variance, given the number of bats that we've sampled. And we find 63 SNPs, shown here in green, that at a false detection rate of 20% seem to have evidence of being under selection, not being consistent with what we'd expect just from neutral drift. So we can then look at the allele frequencies across the genome for the early samples and the late samples. And for just randomly selected SNPs across the genome, we tend to see they're either at very low or very high frequencies, which is what you'd expect for most SNPs across the genome. But for the SNPs in particular that appear to be under selection, they started at slightly higher frequencies in the early samples, and in the later samples have shifted all the way up to not all the way to fixation, but to quite high frequencies. And what this is suggesting is that we're not looking at novel mutations. Instead, we're looking at selection on standing variation. And in addition, these aren't complete sweeps. They're incomplete sweeps, not all the way to fixation. So we're looking at soft, incomplete sweeps, exactly the kind that's especially difficult to detect if we didn't have these temporal samples. We've also looked at diversity around these SNPs, and it's not particularly low, and other signatures that you might expect from hard sweeps are not apparent in these genomes. So these allele frequency changes also allow us to estimate the strength of selection using an approximate Bayesian computation approach. And we're estimating incredibly high levels of selection, about 0.8. Though on the other hand, considering that 95% of the population died off, this is a really strong cost of selection associated with this as well. So we initially expected that these loci that appear to be under selection would be associated with immune function and indicate some ability of these bats to now be successfully fighting off infection by white nose syndrome. That's actually not what we find. Instead, these alleles seem to be more involved with processes that relate to weight gain, with brain function, and more generally with hibernation physiology and behavior. ADCY3, for example, in humans is associated with a genetic form of obesity. In 13-lined ground squirrels, it's in a pathway that helps convert fat into heat during hibernation. 4 other genes are involved in insulin signaling as well. So our working hypothesis right now is that these surviving bats are putting on more weight in the fall, maybe not waking up as much as well. There's been this observation that some of the survivors now seem to be sleepy bats, and therefore they're not burning through their fat stores despite being infected by white nose syndrome, and therefore surviving despite infection. A lot of details still to work out in terms of how exactly these loci may be involved in the physiological changes and ability to tolerate infection by white nose syndrome. So we started to do look at gene expression in these bats. We've also put temperature loggers to understand hibernation physiology for a number of these bats, and starting to look at allele frequency changes in some of the other species that have been infected by white nose syndrome as well. So I think the take-home message here is that we do see signatures of rapid adaptive evolution in these bats that are affected by a novel disease. So now I want to talk about another big environmental change, but now for a marine species, in this case from fishing. So of the examples of phenotypic change in the wild, all of the most extreme changes have been related to fishing. And that's definitely not a novel idea. In fact, J.B.S. Haldane in 1930 said that fishing could lead to extreme and abnormally fast evolution. And you might say that possibly it has. And from a theoretical perspective, the mechanism seems relatively straightforward. So with fishing, individuals are less likely to survive to old age. So individuals that reproduce earlier and at a younger age are more likely to leave offspring. And then they contribute to the next generation, and that leads to evolutionary change over generations, driving evolution towards reproduction at younger age and smaller size. These changes can also have substantial ecological and economic impacts, including lower population stability, more vulnerability to predation, and overall smaller fishery yield. So there's been a lot of concern that fisheries evolution may be occurring. And experiments in the lab have confirmed that this is at least feasible. A relatively famous set of experiments by David Conover where he and colleagues selected replicate populations of silverside, so a small coastal East Coast fish, selected them either for large body size or for small body size. Very strong selection in the lab that they applied, but they were able to show that, yes, they can evolve larger or smaller body sizes. Nina Therkelton has actually gone back to the freezer, taken the fish that were in that experiment and resequenced them, and is able to show that, in fact, there was genomic evolution that accompanied this phenotypic evolution. So these are the two populations that were selected for large size, the two controls here in the middle, and two populations selected for small size. The y-axes here show a test for allele frequency change. And we see that there, across many locations in the genome, we do see large allele frequency changes across the selected lines. And in terms of direct evidence for genomic evolution, this would be considered a gold standard. In terms of what's happening in the wild, though, the evidence has been much more indirect. The researchers have largely tried to infer whether or not evolution has happened based on the phenotypic changes that have been observed, especially based on the age and size at which individuals mature. And many species of fish, including Atlantic cod, now mature at smaller sizes and younger ages. And in fact, the decline in body size for Atlantic cod has become a textbook example of rapid evolution, even though the evidence has been somewhat indirect based on these phenotypic changes. Just to look at some data, this plot is showing the age at which half the population is mature for two populations, one in Canada, one in Norway, over the last few decades. And we can see this large decline in the age of maturation across these two populations. It's also at a time in which industrialized fishing really intensified on these populations, and there were dramatic collapses to low abundance. Some of the lack of recovery has also been blamed potentially on the evolution that may have occurred. One of the challenges, though, is that phenotypic change, there's always the possibility that phenotypic change is instead plasticity rather than evolution. And in fact, you'd expect fish, in many cases, to mature at smaller size and younger ages if there are fewer of their conspecifics around. So we're at a point where we have, theoretically, it makes sense that there's evolution happening in the wild. I don't think that was me, but maybe it was. There we go. We've got experiments that select this as feasible. We see large-scale phenotypic changes. They all suggest rapid evolution could be happening, but we don't actually have genomic evidence in the wild to test whether this actually is what's going on. So in collaboration with Bastian Starr, a wonderful colleague at University of Oslo, we have samples from these two populations, both in Canada and in Norway. So from 1907 in Norway, as well as 2011 and 2014 samples, 1940 in Eastern Canada, as well as 2013 samples. These are from scales and otoliths that have been collected as part of the fisheries management process. The historical samples are. And these are actually the two populations that I showed you earlier we know are now maturing at a younger age. So we did whole genome sequencing, 8 to 10x genomic coverage across each of 113 specimens, again called probabilistic genotypes. And we have a chromosome scale assembly that we can align the reads against for Atlantic cod. So the first thing is that we just have clear evidence that these samples actually are from the same populations through time. Looking at a PCA, the early and the late Canadian samples grouped together, same for the Norwegian samples. We can also use an approach called admixture to divide the samples into different populations. Each of the individuals I'm grouping here by location and by time, but the colors indicate the boxes that they've been sorted into by by admixture. And you can see that all of the Canadian samples are grouping together shown in yellow, and all the Norwegian samples are grouping together there in orange. The other thing we can do by looking at the allele frequency changes on average across the genome is infer effective population sizes. And in both cases, population sizes are quite large, somewhere around 10,000 possibly for Norway and around 35,000 for Canada. We also don't see any evidence for loss of genetic diversity between the early and the late samples, despite the fact that we know abundance has declined quite a bit. It just hasn't declined to very low levels where you expect genetic drift to be especially important. The main focus, though, was trying to understand whether there are particular locations within the genome that have experienced large changes in allele frequency that would be indicative of natural selection acting on that part of the genome. So here we're looking at FST, so it's a measure of allele frequency change, in this case between the early and late Canadian samples and early and late Norwegian samples. Each dot here is a 50 kilobase part of the genome. There we go. Sometimes it just happens, and sometimes I do it. But we don't actually see any locations within the genome that have had especially large allele frequency changes. There are a few cases here that do seem a little bit large, but these are actually windows in the genome where we have very few SNPs. And actually, the changes that we see appear to be consistent with sampling variation and what we expect from drift in these particular cases. So it's very different from what we're seeing in the lab selected experiments that I showed you evidence from earlier. We could also do this as a statistical test to understand whether this is consistent with drift. And if it's not consistent with a null model of drift, we'd expect p-values up here, so less than 0.05, and we don't see that across either population. We've also done a number of other tests for large allele frequency changes or other signatures of selection, including changes in diversity or linkage or Tajima's D. Looking at shared outlier regions among these two populations, testing individual SNPs against a Wright-Fisher null model, or using a principal components analysis-based test for individual SNPs. The limited evidence for selection tends to be very weak. So there are two possibilities based on what I've shown you so far. One, maybe it actually is phenotypic plasticity, and that's sufficient to explain the large phenotypic changes that have been observed through time. The other possibility is that it's a highly polygenic form of evolution with many alleles changing frequency to very small amounts. We've done a lot of simulation testing of the power of the tests I just showed you earlier. And it's clear that we could detect loci of large effect if they're under strong selection. But if that selection is spread across a large number of alleles, we would not be able to detect it. Turns out, though, there are some methods that can detect polygenic evolution. And because it results in subtle shifts in frequency across many loci, if you have replicate samples, either from the same population through time or from two populations experiencing similar selective pressures, you can look at the covariance in allele frequency changes across those comparisons. Convergent covariance. Convergent correlation, sorry. And we do have the samples to actually apply this method. This was work that was recently led by Brendan Reed, a postdoc in our group. So we can do that by comparing the Canadian temporal changes against the Norwegian changes up until the 2011 sample, or we could do the same using the 2014 Norwegian sample. I'm putting question marks up here because these are a test trying to look for evidence of highly polygenic selection. We can also put together a positive control, in this case, comparing the early to the 2011 Norwegian samples against the early to the 2014 sample. So we expect them to be correlated in that case, the same population. So it's a positive control. And then we can also put together a negative control. So the change through time in Canada versus these two modern samples from Norway. Such a short period of time, it's effectively sampling from the same cohort twice, and we expect any differences in these Norwegian samples just to be sampling noise. So that should be a negative control. And here I'm showing you each of the linkage groups in the cod genome, effectively chromosomes, and then the correlation statistic here on the y-axis, where up is more evidence for selection. And here are the results. So we've got the positive controls in purple. Yes, strongly positive. That doesn't mean anything. It just suggests the method is working as we'd expect. Negative controls seem to be near zero, again, as we'd hope. And the actual tests that we're interested in are consistently above zero across much of the genome. We've also done quite a bit of simulation testing to see if these positive correlations would be consistent with low levels of gene flow. Probably unlikely between Canada and Norway, but maybe it's possible. But we can't explain what we're seeing here just based on low levels of migration. We've also tested whether selection on shared deleterious variants might explain this, and also that doesn't seem likely either. One of the interesting themes in studies recently looking at local adaptation of populations is that portions of the genome in large linkage blocks, like in inversions where there's less recombination, often seem to be very important parts of local adaptation, especially when there's ongoing gene flow. The cod genome does have a number of inversions on chromosome 1, 2, 7, and 12. So we tested whether there seemed to be more evidence for selection inside or outside of the inversions. And there doesn't seem to be any particular evidence that the selection is concentrated on the inversions in the cod genome. In fact, doesn't seem to be present on the stronger signals outside the inversions on chromosome 1 and 7, and fairly similar on inside and outside on chromosome 2 and 12. We've also divided up the genome between the parts of the genome that code for proteins and compared to the rest of the genome. And again, selection doesn't seem to be particularly concentrated within coding parts of the genome. Be interesting to continue this work by starting to look at molecular pathways or functional variants within the genome. These kinds of methods, though, do rely on having large numbers of loci. So this is a very different approach than what I showed you initially, where we're focusing on the individual alleles within the genome. This is, by its nature, highly polygenic. So these findings, in many ways, are actually good news for the future of cod. It suggests that the genetic basis for maturing at a later age and larger size, those variants are still present within these cod populations. So the potential for recovery of these phenotypes in the future. And actually, if we go back and look at these phenotypic trajectories I showed you earlier, in the last decade or so, the cod in Norway have appeared to be maturing at a later age than they were in the mid-1990s. And this is coincident with a time when there was much lower fishing pressure and the population size has recovered quite dramatically in that Norwegian cod population. So in this case, temporal sampling has revealed very difficult to detect because it's highly polygenic selection on cod at the same time that they were heavily fished. So in these first two case studies, we're seeing evidence of adaptive evolution, but I think one of the big challenges next is understand how we generalize across a broader group of species. So that question has taken me to two very different places. One of which is the Philippines. It's a place I've been working for the last 15 years. It's a place of incredible biodiversity, both on land and in the ocean, but also very intense human impacts. We've got 100 million people living across 7,500 islands. In many ways, quite an urbanized ocean. It's been called a hotspot both for biodiversity as well as threats to biodiversity. The other place it's taken us is to the Smithsonian. and the National Museum of Natural History, which has one of the largest collections of fishes in the world, many of which were collected by what was called the Albatross Expedition, which went to the Philippines in 1908 and 1909 as quite a colonialist scientific expedition just after the Spanish-American War. They collected 79,000 specimens over that expedition. And one of the interesting quirks of this particular expedition is that they stored the fishes in sugarcane alcohol, effectively high-proof rum. Normally, you'd store fish in formalin. Formalin tends to degrade and chop up DNA. That makes it very difficult to sequence. Ethanol does a pretty good job of preserving DNA. So in collaboration with a wonderful set of Filipino colleagues, including Aniel Alcala, who unfortunately just recently passed away, we've been redoing many of the albatross collections so we can start to understand evolutionary change that's happened over the last century. We're using shotgun sequencing to build draft genomes for many coastal fishes, and then either a bead-based capture method or low coverage whole genome sequencing to reconstruct allele frequencies in the historical and the modern samples. And we're doing this not just for a single species, but actually across more than 20 coastal fishes. Everything from Sprataloides delicaculus, with a generation time of only a few months, up to species like Celarius fasciatus with a generation time 25 times greater, as well as other species like Geras oyena with a relatively large body size, but relatively short generation time. And so we're hypothesizing stronger genetic drift in species with shorter generation times because we've got a greater number of generations that have passed over this period of time. We also are hypothesizing stronger drift in species with larger body sizes. They tend to have lower abundance, lower population sizes. And what's so unique about this particular study, and what makes me so excited about it, is that it's a fairly comprehensive look across a number of species, all from the same location over the same period of time. So just to show you one example, this is what's called a joint site frequency spectrum, and it records the changes in allele frequencies across many SNPs within the genome. In this case, for this species, Equileides laterofenestra. We thought this was a different species until we genotyped it, and then we realized it was not the species that had been identified initially. It's been interesting as that happens across a few of our samples. But we've got allele frequency in the historical and in the contemporary. And from this joint site frequency spectrum, we can reconstruct the demographic history of this population. And one of the things we see is a dramatic decline in effective population size over the last century, coincident with a time of rapidly growing human impacts on the ocean in the Philippines. As we start to do this across a larger number of species, I'll be able to tell you how common this kind of demographic history is across the range of species that we're sampling. There's also a strong capacity building component to this project. We're running bioinformatics workshops in the Philippines with both Filipino and American students, and as well as some faculty. We have some Filipino students working directly on the project as well. It's also a component of this project that provides research experiences for undergraduates, American undergraduates that have been going over to the Philippines. Marielle Malabag in particular is quite close to submitting a paper on genetic diversity and how it differs across a wide range of fish life history types. And more broadly, my goal across the next few years is to combine both ecoevolutionary theory and temporal population genomic methods to understand how evolutionary change differs across species of a variety of life history types. For which species is evolutionary rescue likely to be a viable response to ongoing global change, and for which species is genetic drift going to be a dominant force that then leads to loss of diversity and potentially even an extinction vortex. I think to be able to generalize this needs to be based on a trait-based approach, though I think aspects of the environment may also be quite helpful in understanding which species are likely to have the functional genomic diversity to adapt to ongoing change. So just to summarize, you've seen temporal genomics suggesting strong selection for hibernation-related traits in bats infected with a novel disease. Temporal genomic approaches also revealed that rapid phenotypic changes in Atlantic cod appeared to be associated with highly polygenic evolution, very subtle frequency changes that would be hard to detect with many traditional methods for detecting selection. And finally, ongoing work to understand evolutionary changes across a wider diversity of species. And I guess more generally, I'd argue that these temporal methods provide a powerful window into understanding evolutionary changes in the wild, and that many of these dynamics actually would be invisible if we didn't have this explicitly temporal sampling. We're starting to see how selection, and in some cases, especially the bats, high mortality rates really can lead to strong selection. And finally, I think it's, more broadly, it's important to integrate across both evolutionary and ecological changes to understand how these ecological systems are adapting to rapid environmental change, how ecological and evolutionary forces may play off against each other, and then so we can better understand how to apply those lessons to conservation and ongoing management as well. So with that, wonderful set of colleagues to thank, folks in my lab that do a lot of this work and make this work so rewarding and so much fun, and the funders that make all of this possible. So thanks to all of you for listening.